My name's Associate Professor Darren Saunders from UNSW, and I'll be your MC for the formal part of this evening's proceedings. It's now my very great pleasure and privilege to introduce you to tonight's guest speaker, Dr Kara Vine-Perrow. Dr Perrow is a group leader of the Targeted Cancer Therapeutics Research Laboratory. She's the co-founder of the Cancer Drug Discovery Group at the Illawarra Health and Medical Research Institute at UOW. She received her PhD in cancer cell biology at UOW in 2007 and has trained internationally in cancer biology and metastasis at the Finsen Laboratory in Copenhagen and at the University of Malmo in Sweden. And she has more than 10 years of experience in the field of preclinical drug development and delivery and has made significant contributions, along with academic and clinical co-investigators, to the development of an all-in-one chemotherapy formulation that has just completed phase 1/2 clinical trials in the Illawarra, which is quite an achievement. In the last 5 years, her contribution to the field of drug discovery and drug delivery has been recognised by a number of awards and prizes, including a UOW Vice-Chancellor's Postdoctoral Fellowship, Dr Margaret Gardner Medical Research Scholarship, and an Australian Academy of Science Pathways Travel Award. Please join me in welcoming Kara to the stage.
Wow, what an introduction. Thank you so much, Darren. Thanks, Alison, for that kind introduction. And thank you all for coming out tonight and supporting this talk and my research. And I want to echo Alison's comments and just express how grateful I am for all of the community support, because we absolutely need it for our research, and it keeps us motivated and it keeps us going. So thanks. This is me. It's a great pleasure to be kickstarting the first Alumni Knowledge Series talk for 2020, and I'm really excited to share with you some of the research that my team is doing to develop new treatments to outsmart cancer. I'm not actually going to be talking about nanotechnology tonight, so sorry to disappoint you, but if you want to ask me about that afterwards, I'll be happy to talk more about it. But before I do talk about my research, I thought we should start at the very beginning. So this may surprise some of you to know, but cancer is not a new disease. It's a disease that spans the breadth of human existence. So what you're looking at here is a fossilised toe bone from a 1.7 million year old ancient human ancestor that has cancer in it. Now this fossilised toe bone was discovered in South Africa from the Cradle of Humankind World Heritage Site near Johannesburg. And it was probably from one of our ancestors, one of the hominids. So probably either Homo ergaster or Australopithecus robustus. And the data that was published in the South African Journal of Science described this cancer as osteosarcoma, which is a rare and deadly form of the disease. But this isn't the first time that cancer's been discovered in remains before. We've discovered cancer in a Croatian Neanderthal who walked the earth over 120,000 years ago. Similarly, we've discovered cancer in the ancient Egyptian civilization. So what you're looking at here is a CT scan, just here, a CT scan of the lumbar spine region of a 2,200-year-old Egyptian mummy. And these white lesions here represent the oldest case of metastatic prostate cancer. So cancer has manifested itself throughout human history. It's affected people of all ages. It's basically caused suffering on a global scale. In fact, 1 in every 6 deaths is due to cancer. 1 in every 6. This makes cancer the second leading cause of death worldwide, second only to cardiovascular disease. And you can see here, with 9.6 million deaths from cancer each year, that it kills more than respiratory diseases, dementia, and infectious diseases such as tuberculosis, HIV, AIDS, and malaria, all combined. And with an ageing population and an increasing population, the incidence of cancer is only going to increase. But it's not all bad news. Cancer death rates are actually going down. Death rates being the number of deaths from different types of cancer per 100,000 individuals, and that's what you're looking at here. So number of deaths per 100,000 people over time based on cancer type. And in fact, we've seen a 25% decrease in cancer-related deaths since the 1980s. So what this is telling us is that scientists and clinicians, we're working together and we're making progress. We know more about cancer than we ever have before, but we've still got a lot more to learn. So what is cancer? Cancer is a broad term used to describe over 200 related but distinct diseases in which cells divide out of control and they invade nearby tissues. And in some cases, these cancer cells can access the blood vessels and the lymphatic system and spread to distant sites in the body. And this is known as metastasis, and it's the most lethal form of cancer, and it's responsible for over 90% of all cancer-related deaths. So how does it start? Well, we have more than 1 trillion cells in our body, and each of these cells has a specific function and a fixed lifespan. Cancer starts with the accumulation of mistakes or mutation in the DNA of just a single cell or just a few cells. Now, DNA is a long string of molecules that we call nucleotides, which can be represented here by the letters A, T, G, and C. And the way that these letters come together form a gene, essentially. And this gene instructs the cell how to behave and how to function. And one small change in these letters, one tiny mistake, an A to a G, for example, can change the way that gene instructs the cell to function. And this can have detrimental effects on the behaviour of the cell. So mutations can be caused by a number of factors. They can be spontaneous, that is, that they can arise during cell division, whereby the DNA is replicated to pass on to its daughter cells, and I'll talk a bit about that in a moment. But they can also be caused by the environment, so exposure to tobacco smoke, for example, viruses, UV radiation. So as I said before, as cells divide, they make copies of their DNA, and they pass that on to their daughter cells, as you can see up here. When normal cells divide, they only do it when they're required. And that's to maintain the number of cells within a tissue or an organ so that it functions correctly. In normal cells, if the DNA is damaged, the cell has a mechanism to detect that, and it starts to initiate a process of cell death that we call apoptosis. And it does this to prevent that damaged DNA being passed on to its daughter cells. However, cancer cells don't respond normally to these control mechanisms. And once that damaged DNA is passed on to its daughter cells, those daughter cells can go on to divide and also accumulate mutations in the DNA. They can then go on to acquire a second mutation, a third mutation, a fourth mutation, and so on, and overall this leads to unregulated growth. So we've learned a lot about cancer by looking at cells under the microscope, studying their growth properties outside the human body. So when a normal cell undergoes cell division, as I said, it only does it a finite number of times. And so if we take normal cells outside of the body and we culture them, we follow that they undergo a certain number of divisions and then they stop dividing, which is represented here by the green line. They might even then go on and die. But if we take cancer cells or cells isolated from a tumour and put them under the same condition, they will continue to divide indefinitely. And this is a hallmark of cancer, and it's known as immortality. So now I'm going to ask a question. Has anyone read the book or seen the movie The Immortal Life of Henrietta Lacks? A few of you. That's great. It's a fascinating story. So if you haven't read it, I highly encourage you to. So for those of you who don't know, Henrietta Lacks was a poor tobacco farmer from Baltimore, and she had her cervical cancer cells taken from her without her knowledge. Now, this is a really sad story because she was never recognised for these cells, but these cells contributed to many breakthroughs in the field of medicine. For example, by using these cells, which we now term HeLa, so he coming from the first part of Henrietta's name and La coming from the first two letters of her last name, have contributed to the development of the polio vaccine, for example, and further understanding things like in vitro fertilisation. Now, even though Henrietta died the same year that her cells were taken and cultured outside of her body, those cells still exist today. Over 50 years later, those cells are still dividing in laboratories around the world, including mine. They've undergone over 20,000 cell divisions. So we've heard that the accumulation of genetic mutations to the DNA causes cells to grow outside of normal control. But that's not all to this sort of multidimensional cancer story. So just like you and I respond to the environment around us, cancer cells also respond to their environment. And we call this the tumour microenvironment. And what this is, it's a specialised niche that surrounds the tumour that consists of normal host cells, so normal cells within our body, that the tumour cell recruits towards it. And these can be immune cells, for example, macrophages and lymphocytes, fibroblasts, which are a cell type that's known to lay down the structural framework of tissues and glue cells together, as well as blood vessels. And what happens is that cells, these tumour cells, which are shown here as the brown spots or round circles, they're constantly communicating to these other host cells within their microenvironment. They're sending out signals, for example, to tell the blood vessels to grow towards it so that it can obtain nutrients. And this is also the way that cancer is able to hijack these blood vessels and move throughout the body and metastasise, like I mentioned earlier. They're also able to send out signals to suppress the immune system. So switch the immune system off when it would normally otherwise recognise it and destroy it. So cancer is complex. However, our arsenal of drugs available to treat cancer are rapidly growing. For example, we have local therapies such as surgery and radiation, or we've got systemic or whole body therapies such as hormone therapy, chemotherapy, immunotherapy. And the more we understand about the biology of cancer and understand those genetic mutations and how they drive the cancer, we're able to generate greater precision medicines that target those particular aberrant pathways. And these targeted therapies, as we call them, tend to be more effective and less toxic than cytotoxic chemotherapies. So this has really revolutionised the way that we treat cancer. However, cancer cells are smart. Cancer cells basically evolve or adapt to those treatments and survive even in the presence of those drugs or therapies. So resistance to therapy is an increasing clinical problem, and it's responsible for a large number of patient relapse. So as you can see here, 30 to 50% of non-small cell lung cancer patients relapse and die post-surgery because of resistance. 50 to 70% of ovarian adenocarcinomas reoccur within 1 year after surgery and associated chemotherapy because of resistance. And 20% of paediatric acute lymphoblastic leukemia patients develop recurrence even after an initial complete remission because of resistance. So resistance can be innate, that is that it exists in the cells already, and basically as someone is exposed to the therapy, they don't respond. Or it can be acquired over time, and that is that a patient can initially respond to a drug, and then the cancer cells evolve and then later relapse and not respond at all. And the mechanisms that underpin this resistance are really varied and diverse. But one thing we do know is that heterogeneity or diversity in cancer is a bad thing. So when we think about diversity, generally we think that diversity is a fantastic thing. In the context of cancer, diversity or heterogeneity is bad. And so, apologies if you can't see this here, but here we've got 3 figures with different coloured spots in them that each represent a different tumour. So we call this intra-patient variation. And this means that even if patients present with the same type of cancer, they can respond to treatment differently. We also see intra-patient variation, and that is if a patient has multiple tumours within the body, those 2 tumours might progress or respond to therapy in different ways. And similarly, if we hone in on just one of those tumours and have a look at the cell types that make it up under the microscope, we can see here that each individual colour of these cells is indicative of different mutations from which it arose. So this means that even cells within a single tumour respond to therapy different and progress the disease in different ways. And so this is where my research comes in. So what we are trying to do is to understand these resistance mechanisms that provide a barrier to effective drug delivery or effective delivery of drugs to cancer cells, and try and outsmart it and think about new ways that we can overcome it. And so in the interest of time today, I'm just going to give you 2 examples of how we're working to outsmart cancer. So in the first example, I'm going to be talking about how we're overcoming resistance to multiple drugs, otherwise known as the multi-drug resistance effect. So multi-drug resistance is a major clinical obstacle to the treatment of cancer. Multi-drug resistance occurs when cancer cells become resistant to a variety of structurally and functionally unrelated anti-cancer agents, even if the patient hasn't previously been exposed to that drug. And so what I'm showing you up here is a number of different anti-cancer drugs, and I want to highlight the differences in their structure. So a patient might receive vinblastine, for example, and initially respond. After a period of time of receiving that treatment, that patient may no longer respond to vinblastine, but then when we move to the second line of treatment, as shown here, any one of these drugs, daunorubicin, for example, the patient doesn't respond to that drug either, even though it's structurally different and has a different molecular target within the cancer cell. So one of the, best studied and well-known driver of multi-drug resistance in cancer is a protein that exists on the surface of cancer cells called P-glycoprotein. And what you're looking at here is the outside of the cell, the inside of the cell, the cancer cell membrane, and this P-glycoprotein spanning that membrane here. How we can think about this P-glycoprotein is that it is acting as an efflux pump or a molecular vacuum. And what this means is when the drug comes into contact with the cell, moves through the cell membrane inside the cell where it would normally interact with its cellular target and kill the cell, before it can do that, it's detected by this efflux pump and quickly shuttled straight back out. What this means is the drug can't accumulate to the toxic concentrations it needs to kill the cell, and the cell continues to divide and grow even in the presence of that drug. Now, one of the ways that scientists have tried to overcome this PGP-mediated efflux is through the co-administration of chemotherapeutic drugs and PGP inhibitors. So what would happen here is that these drugs are co-administered, they pass through the cell membrane, these PGP inhibitors interact with this efflux pump and switch the vacuum off. This therefore allows the chemotherapy to accumulate to toxic concentrations and kill the cell. In theory, this should work really well. And in fact, in a lot of the cell-based studies and the preclinical models that we use, it works fantastically. However, when we try and translate that into patients, we don't see the same level of effect. And this is because these inhibitors are not specific enough for inhibiting just the cancer cell P-glycoprotein. In fact, they hit all other efflux pumps that exist in the body, because efflux pumps actually have a role in maintaining normal physiology. They also hit other ion channels, and what this does is creates very highly toxic side effects for the patient. So a second way we can think about overcoming this P-glycoprotein pump is to design new drugs that aren't recognised by the pump at all. Essentially, they work as stealth compounds. So they move through the cell membrane into the cell and they're not recognised at all. And that's what I'm going to be talking to you about in a moment. But before I do, I just wanted to highlight that P-glycoprotein is a clinical marker of disease progression. And what I'm showing you here is a survival curve of patients with acute lymphoblastic leukemia. And what we can see is that those patients who have high PGP expression on the surface of their cells tend to do poorer over time than those patients who have low PGP expressed on their cells. And this is just a tissue section from liver cancer showing specific P-glycoprotein staining in brown. And as you can see, this P-glycoprotein, as I said before, is concentrated on the membrane of those cells. So I'm glad John Bremner's here tonight, because I want to talk to you about a class of compounds that John and I discovered back in 2006, 2007. So we discovered a class of compounds, but we originally isolated the initial sort of parent structure, which I've shown here in blue, from the egg masses of a marine mollusk known as Dicotheus orbiter. We extracted these compounds out, and we identified that these compounds actually had quite potent activity against cancer cells, but they were less toxic against normal human cells. We then went on to, as John Bremner is quite a well-known medicinal chemist, we went on to structurally derivatise these molecules and try and increase their potency. And what we did was added these groups here at the N1 position. We made these N-alkyl and N-aryl derivatives, and what this did was it increased the potency over 1,000-fold. So now we had quite active compounds that were effective at killing cancer cells and selective for cancer cells over normal healthy cells. And along the way, we also discovered that these drugs were not substrates for PGP. So they weren't recognised and they weren't shuttled out of the cell. And I'm just going to tell you how we went about doing some of those studies. So we used 3 different cancer cell lines, one that had low PGP expression on its cell surface here, and then 2 that had intermediate and high expression of PGP on its surface. And so we termed these a drug-sensitive and a drug-resistant cancer cell line. And when we expose the sensitive cells to a conventional chemotherapeutic drug, like doxorubicin, which is currently used in the clinical management of breast cancer, for example, we saw what we expected, and that is that this doxorubicin had an effect and killed these cancer cells. When we exposed this drug to resistant cells, however, we didn't see the same cell-killing properties. These cells were not responsive, they were resistant. However, when we exposed our N-alkylicin to these resistant cells, they killed the same proportion of resistant cells as the sensitive cells. So this was really exciting. And here's some of the data here. So what you're looking at here is cell viability on the y-axis and increasing drug concentration on the x-axis. And you can see that as we increase the drug concentration, we're reducing the viability of those cancer cells. Most importantly, the red line indicates the drug-resistant cell line, and the black line indicates the drug-sensitive cell line, and both of them behaved in a very same way. They responded to the drug treatment in the same way. Compare that to doxorubicin, for example, and you can see that we require much, much higher concentrations to kill those cells. And in fact, that effect that we're looking at there for doxorubicin wasn't unique for that drug. We also saw that for other anti-cancer drugs, such as the Vinca alkaloids, paclitaxel, colchicine, 5-fluorouracil. And this is a hallmark of multi-drug resistance. So we were interested to know whether or not our drug maintained its mechanism of action in these resistant cells. And so the way that we did this was just using a simple microscopy experiment. We looked at the morphology of a lymphoma cell line, a sensitive and resistant line under the microscope. And you can see here with no drug treatment, the morphology is the same. They're nice and round. When we expose the sensitive line to the vinblastine, which is an anti-cancer drug that targets the cell's skeleton, essentially, or microtubules, we saw a characteristic change in the morphology, as you can see here. And this change represents the first part of a series of events that essentially leads to cell death. However, we didn't see the same morphology in the resistant cells. This class of drug was ineffective. When we exposed our sensitive and resistant lines to the N-alkylicins, we saw this same morphological response, which told us that the N-alkylicins were interacting with their intracellular target, which is the same as vinblastine, targeting the cell skeleton in exactly the same way, but it was bypassing that PGP efflux pump. And we have a few ideas about how it might be doing this. Potentially, it is entering, because it's quite a lipophilic drug, it's entering the cell very, very rapidly, too rapidly too fast for the efflux pump to be pumping it out. An alternative hypothesis is that because this is a lipophilic drug, it's actually interacting with the cell membrane and changing the lipid rafts that hold that P-glycoprotein in the membrane such that it has an effect on the P-glycoprotein structure. Now, another interesting finding that I want to talk about is that the N-alkylicetins didn't seem to have this off-target neurotoxic effect that vinblastine and other drugs do. And I'll explain. So vinblastine, as I said before, is an anti-cancer drug that targets the cell's skeleton, the microtubules within that skeleton. We also find these microtubules in peripheral neurons, so sensory and motor neurons throughout the body. And actually, chemotherapy-induced peripheral neuropathy, or damage to these neurons, is a hallmark or an associated side effect of vinblastine therapy. And it can be mild, for example, just cause tingling or pins and needles sensation, or it can be quite severe to the point where people have pain, burning, numbness, loss of motor function, but also to a point where you have to reduce the treatment because the side effects are too great. And this is particularly a problem in children who are receiving this kind of therapy. So we wanted to assess whether or not our N-alkylicetins, who have the same mode of action as this drug, equally had a neurotoxic effect. And what I'm showing you here is some neurons that we have in culture. So the cell body here is green, and the neurites that are coming out are orange. And this is what a healthy cell looks like. When we expose this drug to vinblastine, we see a significant reduction in the number of neurites that are coming out of that cell body, and that indicates neurotoxicity. When we look at the effect of the N-alkylicetin at the same concentration, the same drug exposure time, we see a morphology that's identical to that of the control. We've also collaborated with electrophysiologists, Professor David Adams's group in IMRI, to also study the cell function, so looking at the electrophysiology, and we see a similar trend there as well. So where to from here? Well, we need to formulate these small molecules so that they're safe and suitable for injection into humans. And we're currently working on that now. In fact, I will talk about nanotechnology because the way that we're doing that is encapsulating these into small, tiny little particles that mean that we can administer these drugs at physiological pH directly into the bloodstream. We're also further testing their neurotoxicity profiles in other more advanced models of neuropathy. And in the future, what we think is that we could use these drugs in 3 different ways. The first could be as a second-line therapy in patients who have relapsed to first-line therapy, so coming in as a second agent when the cancer becomes resistant to the first. The second approach could be that we use these drugs as a new standard of care, a completely new agent that's used as a first-line therapy. Or more realistically, we would probably use a combination of this drug and the currently available drugs together to try and stop or reduce the development of multi-drug resistance in the first place. I'm going to change it up a little bit now, and I'm now going to move on to our example 2. And I'm going to be talking about how we're trying to overcome the tumour microenvironment in the context of pancreatic cancer. So many of you probably already know these statistics, but I'll tell you anyway that pancreatic cancer is one of the most lethal solid malignancies. Less than 1 in 10 people survive 5 years after diagnosis. And you can see here that globally the incidence is almost equal to its mortality. And this statistic really hasn't changed much over the last 40 years. And to highlight how dismal this is, I want to compare this to breast cancer, for example, where we see 91% of patients still alive 5 years after diagnosis. And although the incidence is increasing, likely due to an increasing and ageing population, the mortality is actually decreasing. So what this highlights to us is, although pancreatic cancer is rarer than breast cancer, we still don't understand the biology of the disease enough. And we need to further understand this so that we can design new and better ways to treat it. So how do we treat pancreatic cancer? Well, surgery, chemotherapy and radiotherapy are all the current treatment approaches that we use to control the disease and alleviate the symptoms of the disease. However, they rarely produce a cure. In fact, surgical resection of the tumour, so the physical removal of the tumour, is the only potential chance of cure, but less than 20% of patients are eligible to undergo this surgery because their disease is too advanced at the time of diagnosis. So why is pancreatic cancer so deadly? What are these barriers that we need to overcome? Well, a hallmark of pancreatic cancer is this dense fibrous tissue or scar-like tissue that surrounds the tumour. And you can see that here in pink. And this is essentially the tumour microenvironment. And what we find in this dense scar-like tissue are a number of cell types, including those fibroblasts that I talked about earlier. So an increased number of fibroblasts, which are responsible for the increased deposition of these structural components, these extracellular matrix components in and around the tumour. So deposition of proteins like collagen, like hyaluronan, and altogether what this does is it increases the stiffness of the tumour. And in fact, I think this particular desmoplastic reaction or fibrosis contributes up to 80% of the tumour mass. So it's a significant, has a significant impact. And altogether, as I mentioned, this fibrotic reaction increases the tissue stiffness. It increases the interstitial fluid pressure. So we've got higher fluid inside the tumour than outside. And it compresses the limited blood vessels that supply the tumour. And what this means is that it's very, very difficult to get systemically administered chemotherapeutic drugs at high enough concentrations into the tumour to have an effect. I guess it's not as easy as just dosing up the patient with higher concentrations of drug because of the dose-limiting toxicity. We just simply can't get enough drug to the tumour where it's needed. So how has my research group trying to overcome these barriers? Well, what we've done is we have worked with some of the clinicians at Wollongong Hospital and worked with materials scientists at the Innovation Campus here in the Intelligent Polymer Research Institute to design a polymer implant, as you can see here, that contains two chemotherapeutic drugs. So these two chemotherapeutic drugs, gemcitabine and paclitaxel, are already used for the treatment of pancreatic cancer, but they only moderately increase survival because of the phenomenon that I spoke about before. And so we've loaded these two drugs, gemcitabine and paclitaxel, into these polymer-based implants. And the reason we've done this is so that we can insert this implant directly into the tumour where it's needed. So essentially this implant acts like a Trojan horse, and the idea is that it kills the tumour from the inside out. It doesn't have to bypass that physical structure. We've designed these implants to elute the drug over a period of 2 to 3 weeks with the aim of killing the tumour cells, but because it's locally delivered, should induce minimal healthy tissue toxicity. And then because of this localised approach, as I said, the drugs can be delivered at much higher concentrations than we could get via systemic administration. And we've designed the implants to contain echogenic particles that mean that they're visible under ultrasound, as you can see here. And the reason that this is important is so that we can use a minimally invasive gastrointestinal procedure to deliver them, known as endoscope ultrasound-guided fine needle injection. So what this means is that we can load the endoscope needle with our implants, then pass the echo endoscope along with the needle through the patient's mouth, down the esophagus, and into the stomach. Once we reach the stomach, we can pass the thin needle a short distance through the stomach wall and deploy the implant directly into the tumour. This is a minimally invasive procedure, and this is where we're aiming to take this technology. So how we envisage this therapy working is that the surgical implantation of our drug-eluting implant, which is capable of delivering these 2 chemotherapeutic drugs, will achieve tumour control. It will shrink the tumour, as I said before. And the idea is not to cure the disease, instead to convert non-resectable cases to resectable cases to try and improve the overall survival. So how do we make these implants? Well, we make them right here on Innovation Campus. So we use a one-step wet spinning technique. So we take liquid polymer with our chemotherapeutic drug co-dissolved, and we inject that through a custom-made spinneret into a coagulation bath. When the polymer hits the coagulation bath, it spontaneously cross-links and solidifies, forming a continuous fibre, as you can see up here, with a core shell morphology. So what this means, what I'm showing you here, is a cross-section through our implant using scanning electron microscopy. And we can see here that we have a core and a shell. And the reason we've designed the implants to have this morphology is so that we can load 2 different chemotherapeutic drugs that have 2 different chemical properties. One's hydrophobic, one's hydrophilic. And this allows us to administer the drugs in the same implant at the same time. And we can modify the release of the drug from the implant by doing things like changing the thickness of this shell, changing the pore size, for example, and the cross-linking density. And we've done that. And this is just a graph to show you that by changing different parameters of our implant, we can move from this blue line here that shows a burst release of the drug. So this is drug released over time. We're seeing a more burst release, and we can change a few of the properties of our implant to obtain a more sustained release of the drug. Now, in order to test the effectiveness of our implant, we need to use model systems that better model the disease. And so in my lab, we've developed a 3-dimensional cell culture method to make 3-dimensional pancreatic spheroids. So you can consider these as tumours in a dish, essentially. And these are a really nice intermediate between the oversimplified way that we grow cells in a 2-dimensional culture, just as a flat monolayer, and the more expensive, complex and ethically restrictive animal models. What's nice about these tumours in a dish is that they accurately, to a degree, recapitulate the tumour that we find in the human body. So we see nutrient, oxygen and waste gradients, we see tight cell-to-cell interactions, we see chemoresistance. And we see the organisation of cells in an architecture that are similar to that in the body. For example, we have a zone of rapidly proliferating cells on the outside, and this is reflected here by this green signal on our pancreatic spheroid. So now that we have these spheroids as a model, we can use them to assess the ability of our implant to deliver the drug to them. And so, as I said before, one of the main barriers that we're trying to overcome is the limit that the dense stroma places upon us to deliver high concentrations of drug to the tumour. So we're looking at trying to increase the accumulation and penetration using our implant. And so what we've done here is we've taken these tumour spheroids, and in the solution around it, we've added free drug. We've added a fluorescent drug called doxorubicin. And then at various time points, we've imaged this spheroid, but we've washed the solution that surrounds the spheroid at different time points to simulate clearance from the body. And you can see here, we've got a small amount of uptake into the spheroid. I don't know if you can see, actually. This little red spot here that largely remains static over time. It's taken up and it doesn't change. When we use 10 times the free drug, we see a greater uptake initially. Again, it remains static over time, but there's a few things I want to point out. The first is that it's taken up heterogeneously throughout the spheroid. The second is, at the later time point, we can see this drug moving out of the spheroid down its concentration gradient back into solution. So we're losing the drug in the tumour. When we deliver our drug from our implant, we see something quite different. We see an initial uptake, but this uptake is actually linear over time, and we see at the end of this particular experiment significantly greater drug associated with this spheroid, and it is distributed throughout that spheroid. And this is because the implant is acting as a drug depot and is constantly releasing that drug over time. Similarly, we can assess the depth of penetration of our drug into these spheroids. And so we use confocal scanning laser microscopy, and we take images at different depths throughout that spheroid, and we quantify the amount of fluorescent signal or drug, and we can determine how far it's penetrated in. And again, you can see that when our drug is delivered from our implant, it penetrates further than if we put it in free in solution. And what we think is happening here is two things. One is, as I said before, the implant is acting as a drug depot and is constantly releasing the drug over time, which means that there is more drug available to the spheroid. The second is that we think that the polymer that we use to make this implant actually has another function. It's actually acting as a permeation enhancer. That means that it's basically disrupting those tight cell-to-cell junctions between those cancer cells and allowing the chemotherapeutic drug to penetrate through that gap junction and perfuse throughout the tissue. So we next wanted to move to a more complex model and assess this effect. And so here we used a mouse model of pancreatic cancer. And so what we did was we injected our pancreatic cancer cells into this mouse, allowed the tumours to grow to a certain volume, and then once that tumour had grown to a certain volume, we either delivered our drug-loaded implant directly into the tumour, or we delivered an empty implant and gave the same amount of drug, but systemically, so via the tail vein. Then we basically waited 24 hours, and then we harvested that tumour and we quantified the amount of drug that was in that tumour. And we can see here, the red is indicative of our drug, that we had about 10 times the amount of drug in the tumours of mice that received the drug-loaded implant compared to when the same drug was delivered systemically. So again, we're overcoming this barrier to effective delivery. We can also use these tumours in a dish to assess the efficacy or anti-tumour activity of our implant. And so here what you're looking at is spheroids that have been incubated with just an empty implant, so no drug. And what you're seeing here is that the spheroid continues to grow, increases in diameter over time. And when we compare this to the no treatment control, we can see that they're growing in the same way. And so this indicates to us that the implant is not causing any non-specific toxicity. When we load the implant with our drug, we can see straight away by day 5 that we've stopped the spheroid from growing. And then when we continue the experiment out over time, we're starting to destroy the architecture of that spheroid. Again, we can assess the efficacy of our approach in a pancreatic cancer animal model. And here, we've injected our pancreatic cells into the flank of the mouse. We've allowed that to grow to a certain tumour volume to represent locally advanced disease. And then we've inserted our implant. In this case, we've inserted it, because of the implant size, immediately adjacent to the tumour. And then we've monitored tumour growth, weight and behaviour over time. And what I want to draw your attention to down here is this red line. So this red line is the tumour response in mice that received our dual drug-loaded implant, so our gemcitabine and paclitaxel together. And what I want you to compare it to is the green line here, which is the empty implant, but had the same concentration of those two drugs delivered systemically. Importantly, what I want to also point out is that none of the mice lost weight, which is a surrogate marker for toxicity. We saw no adverse effects with our implant, no inflammation. The implant didn't migrate over time. Now, despite this impressive result here, by the end of the study, we didn't see a significant increase in survival, and we think we know why. And that's because when we harvested the tumours and took back these implants that had been in the mice, we found that there was still drug inside those implants. And so I think what we're seeing here is an initial effect, but if we had have continued the study further, we may have seen a more pronounced effect because all of the drug hadn't been released. So we can go back and modulate that release profile, as I said, by modifying the cross-linking density, the thickness of the shell, and other properties so that we can get the right drug release over the right period of time to see a better tumour response. So what's next for our implantable device? So we need to continue completing our preclinical testing using models of disease that actually reflect the clinical disease. That's really important. We've filed a patent and we're in discussions with some potential industry partners to try and drive the commercialisation of this implant. And we've basically demonstrated this implant can really act as a platform technology that allows us to load any drug that we might like into it. So we're looking at loading FOLFIRINOX, which is a 4-drug regimen, into our implants. Now, FOLFIRINOX is used in the treatment of pancreatic cancer, but only in a small subset of patients who are physically fit and are able to tolerate it because it is highly and extremely toxic. So we think that if we can encapsulate this 4-drug regimen into our implant and deliver it locally, we can negate some of those toxic side effects and see an improved tumour response. There's work in the field, and we're also interested in doing this, and that is to co-administer a stroma-depleting agent. So that scar-like tissue that I showed you before that's creating that physical barrier. There's been a lot of work in the field to try and disrupt that through the use of enzymes. And so we're looking at also co-administering a stroma-depleting agent along with our therapy to further enhance that effect. And then finally, we're also looking to immunotherapy. So many of you have probably heard how immunotherapy has really revolutionised the treatment of certain cancers like melanoma, lung cancer, renal cell carcinoma. It's a really powerful approach to harness the body's own immune system, to switch on the immune system again, to recognise that tumour and attack it and clear it. Now, pancreatic cancer hasn't really had much success in that space. And that's because the stroma really is an immune suppressive environment. So there's a few immune cells in there, but not enough for immune checkpoint inhibitors, for example, to work very well. And so we're looking at trying to first stimulate the immune system to bring in those immune cells that are responsible for recognising and clearing the tumour, and then coming in with our immunotherapy to further release the brake on the immune system. So we're really excited about the next steps. So before I leave you with some parting words, I just want to acknowledge my fantastic team. So they're sitting over there, and I'm going to embarrass all of you and ask you to stand up. All of you. Even the new students, stand up. So research is never done in isolation, and I have a fantastic, young, passionate and enthusiastic team behind me, and they have been the ones responsible for generating most of the data that I've presented here today. So I want to acknowledge them. I also want to acknowledge our funding partners, the Illawarra cancer carers and the community for their continued support of our work. They have been absolute cheerleaders of our work, and they keep us motivated and moving forward even when we're having those feather duster days. So just some final thoughts. I really think there's never been a greater time in history for the cancer field. Even though the number of cases of cancer are increasing, the overall death rates are declining, and people are living longer with the disease than they have ever before. With advances in understanding the molecular biology of cancer, using whole genome sequencing, understanding those genetic mutations that are driving disease, we will further identify barriers to effective treatment and move away from a one-size-fits-all approach to a more precision, customisable, personalised medicine approach. As I look into the future, I strongly believe that we have never been in a better position to maintain the positive momentum against cancer. We know more about cancer than we ever have before. And we now have the scientific knowledge, and we've developed new and cutting-edge technologies that give us the capability to deliver the next generation of innovative breakthroughs to outsmart cancer. Thank you.
Wow, thanks, Kara. I'm familiar with a lot of that work, but I've seen it in a new light, and you did an amazing job of walking through a very complex beast and making it easy to understand, so thank you. I've got a few thank yous to make, but first and foremost, please join me in thanking Kara for a fantastic talk.